week 2 of the quarter. Hopefully you made it through the week 1 without any really big issues. I know that week 1 is really kind of always overwhelming for students, so hopefully you made that through. For this week, what we are focusing on is going to be chapters 34, chapter 35, and chapter 42. So for the day 1 video, what I really want to focus on is chapter 34, and then in day 2 video, I'll talk about chapter 35 and chapter 42. So for chapter 34, what this is really focusing on is infection control, which is really going to be the process of reducing exposure to pathogens to prevent the spread of diseases. Pathogens are really just disease-causing microorganisms that are found almost everywhere, in our water, in our food, on our bodies, and the animals, in the air. They're everywhere, okay? As a medical assistant, you really need to be aware of just how easy it is to spread pathogens from one patient to another or one person to another. That's why, as medical professionals, we perform aseptic techniques or use a state of asepsis, which is really just being germ-free and infection-free. And there are 2 types of asepsis that we use. We use medical asepsis and surgical asepsis. Now, medical asepsis is really just referring to being clean and disinfecting and stopping microorganisms once they exit the body. Where surgical asepsis is talking about being sterile and stopping microorganisms before they can enter a body. When we talk about being germ-free and stopping the spread of infections, we really need to understand just how those microorganisms do become spreadable and where they come from and what they are. So there are 4 types of microorganisms that we can come in contact with that can cause diseases: bacteria, viruses, protozoa, and fungi. So those are the 4 big microorganisms that everything can turn into an infection from. And these microorganisms can grow in 2 ways. They can either grow aerobically or anaerobically. Aerobic growth requires oxygen in order to live, where anaerobic growth does not require oxygen to live. So now that we understand how microorganisms get spread and how the infections begin, we need to understand how that process works. How does an infection go from spreading itself? And that's actually going to be used during the chain of infection. So that's how microorganisms get spread. Now, there are 5 links to the chain of infection. The first one being the reservoir host. This is really the beginning. This is where a host body, either an animal or human, actually has that microorganism or that bacteria present within them. Second chain is going to be the portal of exit. This means that the microorganism found in that first host body now wants to exit to another body to infect them. This can go out through any orifice of your body, so nose, eyes, it can go out in your hands, it can go out in through sexual activity, all of those different ways. That's how it can go out of the body. Third link is going to be your means of transmission, and this is really how that microorganism will transport itself through and to another person, either through airborne, droplet, or direct contact with another host. Fourth chain is going to be a portal of entry, so if it got out of a body and now has to get into another body. And the last step is the susceptible host. The susceptible host is just a new host body where that infection can take hold and start reigning on a new person. As a medical assistant, it's really our job to stop that spread of infection and break that chain. And this can be done through proper hand hygiene and hand washing, as well as using the proper protective equipment or PPE. I want you guys to review the procedures in your book 34.2 and 34.3 prior to class this week, because that will actually help you with understanding what we're going to be doing in the lab. So now that you understand how microorganisms get spread and the infection cycle, let's talk about infection control and some of those precautions and standards that are necessary in order to run smoothly in an office. So the first one is the universal precautions. These were created by the CDC in 1985. They were created as a way to protect healthcare employees from patients who have HIV, hepatitis B, or bloodborne pathogens. The theory behind universal precautions was really to treat all blood and bodily fluids as though they're contaminated. Second precaution we're going to talk about is a standard precaution. This was also created by the CDC, and it was created as a recommendation for using and looking at patients regardless of what their disease was. You were going to use these precautions. You want to look and apply these to all bodily fluid, blood, and pretty much anything that comes out of the body except for sweat, whether blood is visible or not. And the last precaution is the transmission-based precaution. This is the second tier, or the second part to the standard precaution. And what it did is it took and it looked at standard precautions and it broke it down a little bit further. It broke it down by how those infections were being spread, okay, or transmitted. And that is through three ways: through the air, or airborne, droplet, and contact. And contact is either direct or indirect. So I physically touch you, or you happen to be in the area. Alright, and the last thing I want to talk about for the video this week is going to be surgical asepsis. So we talked a little bit about medical asepsis in the beginning, and we talked a little bit about surgical at the beginning of the video. And if you remember, I said that's the sterile technique. And that's really done through a three-step process. The first step is called sanitization. And this is the process of cleaning or inhibiting pathogens from growing on equipment or on instruments, and we have to remove that debris. So this happens through rinsing and scrubbing the instruments after they've been used. We're going to do that with a brush, generally a wire brush, and a detergent that has a neutral pH level. After all visible debris has been removed off of the instrument, we're going to rinse that in hot water and allow it to air dry. You don't want to towel dry these because that actually can sometimes put bacteria back into the instrument. So even though the instrument has been cleaned and you see no visible soilage on them anymore, it does not mean that all the bacteria and microorganisms have been destroyed. So now we have to take it to step 2. Step 2 is disinfection. In this step, what we do is we destroy the activity of microorganisms. We use 3 different types of agents during disinfection. We can use germicidal agents or chemical agents, like things such as a bleach solution, 10 parts water, 1.1 part bleach. You can use flowing hot steam, or you can use boiling water. And step 3 is the final step, sterilization. And this is really where we kill all the microorganisms that are left on the body. In this process, we generally use an autoclave. But I'm not going to show you, and I'm not going to tell you how that works. I want you guys to read up on the autoclave. I want you to read up on how long it takes for instruments to be autoclaved, how they work, how you wrap up the instruments for autoclaving, or if that's what you do with them, and also how you load the machine. So please look at procedures 34.5, 34.6, and 34.7 prior to coming to class this week. Alright guys, that's all I have for you, and I will see you all in week 2, day 1. Bye!